Okay, so this is, we're going to take our rope here and attach it to this little plankton net. This is a very small plankton net. So if you were to be working in a marine environment on the ocean, you would have a giant plankton net that would be like handled with electronic winches and huge. But when you're on a tiny little lake like this, you don't need a huge plankton net because there's not as much water to be filtered. So the idea here is that we're going to collect plankton from the water column. We lower this down. We figure out how deep the lake is at this particular spot, and we lower this almost all the way to the bottom, and then we slowly raise it up. And as it comes up, this disc here filters the water through the mesh, which is a very, very fine mesh, so you can collect very small algae and zooplankton, and bigger ones too. And then in this particular setup here, it's collected into a reservoir at the bottom. And the reason this is nice for teaching purposes is we can actually see what's swimming in here afterwards. So we're going to demonstrate how to do that. Okay, so here we have Maddie, who's going to demonstrate how to collect a plankton toe. A plankton toe. She's lowering it down. We've taken the depth here, and it's 7 meters. So she's counting on the rope as she goes. She's just lowering it down.
So, do you think I should stop now?
Yeah, at 7. Okay, and then she's gonna just raise it up sort of steadily through the water column as she pulls the rope in. And then we can calculate the volume of water that's been filtered because we know the diameter of the hole, the diameter of the opening of that net. And then you know the depth that you pulled the water, the net through, and you can calculate the volume of water that was filtered. And then in that way you can calculate the density of different organisms that you collected in the water. But here we're really interested in one thing in particular, which is
the pond fishless, or does it have fish?
yes. Are we collecting fish in our plankton net?
No.
That's not the plan anyways. We're not trying to actually sample fish here. We're just collecting plankton. But we're using them as a clue to figure out whether this lake has fish in it or not. So let's see.
We have phantom midges in here. There's a lot of them swimming around right here. And usually that would mean that it is a fishless pond.
Yes, phantom midges. This one species, we'd have to look in a microscope to identify it for sure, but what it likely is Chaabris americanus. And this is a species that is associated with lakes that don't have fish in them. They swim out in the open water during the daytime, whereas their sister species aren't out at night. They go, or excuse me, during the day. They go down into the sediments during the day. So you wouldn't catch them in a plankton toe during the day. If you went out in the middle of the night, however, in a lake with fish, you would catch the other species. Very cool. So is the lake fishless, we think?
Yes, probably.
Yeah. This is what a Chaabris looks like under the microscope. You might be able to see these darker spots sort of at either end of the abdomen. These are air bladders that allow them to float horizontally in the water column. You can see them here in the container that we collected them in, sort of swimming around. There's a bunch of them in there as they float in the water column, and then they sort of wriggle their bodies to propel themselves through the water. And they are actually predatory organisms. They're part of the zooplankton, but they eat other smaller zooplankton. And so they are able to detect those zooplankton and eat them. And so they play an important role in the food web in the water column. All right, so now we are on Deming Lake, which is a lake that has fish in it, right next to Arco, the one that we sampled with no fish. And we're going to try a plankton toe here and see what we collect. We're using a different net in this lake because when you move between water bodies, you don't want to reuse the same equipment because you might move species from one place to another that aren't meant to be moved around. So we've got a different net. So Maddie's going to lower the net.
It's supposed to be upside down like that.
It's misbehaving. There's some air bubbles coming out of it.
And you're very helpful.
It must be pretty clear. I thought it was closer.
Let's see it. So we're going to see if we catch any cabras in this tow.
And the reason that we know that there's fish on this lake is because we already saw some.
2 sunfish.
Yeah.
1 medium small and 1 small.
We're also noticing a lot fewer bugs on the surface of the lake, maybe because they've been eaten up
It's true.
by the hungry fish.
Do you think I should stop?
How far did you go?
I have no idea.
Maddie forgot to measure the depth of the lake before she did her tow, but that's okay. We just are curious about plankton,
It's
or
experiential
what's in
learning.
there. Yeah, experiential learning. We're just curious about the cabras, so let's see if we got any cabras.
Okay. I'm sorry.
If
I'm
we
a
were
disappointment to
interested
this
in the
family.
density of cabras, we
Oh,
would
you're
need
not.
to know the depth of water that we were measuring. But
But it's not that
we're not being
interesting.
numerical here. We're just
We're
being
not that special.
qualitative in our analysis.
I just want to see what we catch.
She's gonna release whatever's been captured in the bottom of that net with this little clip. We're just doing it into a clear cup here. Did any liquid come out?
No. I'm gonna open it. Holy crap. That's a lot of something.
A lot of something. Is it cabras?
No, it's not. Is it alive? Yeah, it's alive. It's moving a lot.
Wow, there are a lot. So no cabras, but lots of some other kind of zooplankton. But with my 45-year-old eyes, I need a microscope to tell what they are. It's likely that what we captured here are freshwater copepods, which are another type of zooplankton that live in these lakes here in North America.